Hello and welcome back. Raylan and Becca here, your TAs. Today we are going to be doing our organelles test, which Becca will explain further.
So in this organelle lab, Raylan is going to be taking a sample of her cheek cells, and we are going to smear them onto a slide, stain them, and then we are going to be looking for a nucleus underneath the microscope. We are outside starting this today because in order to follow our COVID policy rules, we are only going to be removing masks outdoors. So now, Raylan is going to be removing her mask and swabbing her cheek. You can be pretty aggressive with this. We want to make sure that we get a good amount of cheek cells to make sure that we can see them underneath the microscope. Now, she's going to smear her cheek cells onto her slide, as you can see she's doing. And we'll put our mask back on and head indoors to finish the rest of what we're doing today. Heading right back home. All right, now we're going to get right into this. So Raylan now has her cheek cells on her slide. So the first step, what she needs to do is she is going to stain them with a stain called methylene blue. This stain binds to anything acidic. In this case, we're looking for it to be amino acids, which will be found in our nucleus so that we will see it underneath the microscope. It only requires about a drop or two of this stain. It's very potent, and it should stain our cells quite well. And we're just going to leave that stain on there for about 30 seconds in order to make sure that it goes through the cells and penetrates like we want it to. And your 30 seconds is about up, Raylan, if you're ready to start reabsorbing your stain. So now we are going to be washing the stain out, because if we were to put that under the microscope right now, all you would see is a big blob of blue, which is not helpful. In order to do this, you can see we have our distilled water and our dropper here. And Raylan is going to soak up the stain on one side and add water to the other until there is minimal amounts of the blue stain left. And then you can just hold that off to the side of it even, use the corner, and it'll soak it up. Because we don't want to disturb our cheek cells too much.
Be sure to not get any of the stain on your hands or clothing.
That's Paul you hear. We got him on FaceTime over here.
And be very careful to not get any of this stain on your hands or your clothing, as it is a stain that does bind to amino acids, like we said, which is probably not something that you want to be contacting too much. You might want to get a little bit more of that stain off of there, because that's still going to be quite blue underneath the microscope. Just use your corner. There we are. Another droplet of water and we'll be all set. Now this next step here is another step in our slide prep. You can continue, just talk your way through it. And Raylan is going to take the slide cover she now has in her hand, and she's going to very, very gently place it on top of her slide. In order to do this without getting any bubbles in her slides, so that she can get a better slide picture, she's going to lower it very slowly, as you can see she's doing with her pencil. This is called the pencil drop method. Looks very clear, you can't even see anything on it right now. No bubbles or anything. Good job, Raylan. Now, we're going to bring this over to the microscope and take a look. You can see Raylan has her face shield set out next to our microscope. Again, obeying our COVID policies. While she gets that situated. New face shields are always a little bit tricky there, aren't they, Raylan? You might want to even out your top a little bit. Your forehead padding is a little crooked there for you.
How's that?
Gorgeous. Alright, now that Raylan has this all centered on the microscope, everything's turned on, all set in place on the red lens, which is the 40x. So she is going to get all up in there and focus this until she can see everything in focus at 40x. And since this is the 40, which is the lowest magnification we have on these microscopes, so Raylan could use the coarse adjustment knob if she so chooses, in order to help get it in clarification faster. However, you can see that she's using the fine adjustment, which must mean that she's pretty close. Once she gets this all set in place, she will rotate it up to the next increment of magnification.
Does not want to focus.
The coarse adjustment knob might be a little better.
Possibly. Oh, I think I need to get this closer. Okay. We don't know where it's at. Oh, there we go. I finally see something.
Getting in focus can be a very slow, agonizing, and tedious process sometimes.
But you always got to remember, you will get there. Just have patience.
You just got to keep looking. So now that she was in focus on the 40x, she can rotate it up to the next increment, which here is the 100x, the yellow. So feel free to do that, Raylan.
First, we're going to lower it down.
Oh, you might not want to lower it, as it's going to change your sight picture, and that is the 1000x. You might want to rotate it the other direction.
There we go.
There we go. So typically once you get it in focus, you're just going to want to rotate over to the next increment of measurement, as it'll already be in focus for you. Makes it a touch easier moving ahead, and then you can just continue refining the focus as you go up in magnification.
Alright, and we finally have something here.
Right, now that she's got it at 100, at this point we could go up to the 400, which would allow us to see the cheek cells better. And once she gets it sighted in there, we will be able to show you a couple of pictures that we will just take through the microscope, and we will post up into this for you guys to observe. So we will let Raylin finish out what she's got to do here. Very good job, Raylin. Thank you for the demonstration.
When it comes to microscope drawings, technique is critical. There are a few things that you want to keep in mind when doing these drawings. First, it's more important to draw a smaller area in greater detail than a larger area in poorer detail. So rather than trying to fill in the entire space, I would rather you spend your time drawing a small component of the space in great detail, so that way I can see the different structures that you're trying to label. For example, in this onion epidermal cell, which is labeled below, another critical component of microscope drawings, alongside the magnification that you see, I haven't spent the time drawing all of the cells around it, but instead have done my best to draw the fact that there's a cell wall clearly differentiated from a cell membrane slightly underneath it. To the naked eye, this would look more like a shaded area beneath the cell wall. A cytoplasm as the white space in the cell, and a darker nucleus off to the side. The other thing that you'll notice about the labels is that they all end at the same point. They all come to one side, the right side of the microscope drawing, and that all of the lines are parallel. Each of these features are meant so that the person looking at the image is not trying to decipher which lines are crossed, what is being labeled, or even where the label is. Some of the common mistakes that I've seen in the past have been that folks label them off to the other side. And if I'm looking over here, there's a label over here, I might miss it. Another common mistake is that folks put the label on the inside of the drawing. Again, hard for me to spot. Keep them off to one side, keep them parallel, and do not cross any of the lines. I recognize that we are not all artists. I myself am not good at drawing. My pictures are not pretty, but I can assure you that my microscopy drawings illustrate the point that I'm going for. And that's all that I want you to keep in mind when doing these microscope drawings.
Alright, so now that we've taken these slides under the microscope and hopefully visualized your own human cheek cells, practiced focusing at 100x and 400x, but of course always starting with our 40x magnification, we now have some of these images either on your phone or here on our computers. Let's take a look at what these cells look like now that we know what we're trying to, how to draw and label them. Our human cheek cells should look something like this. Again, a little bit grainy because of the picture quality, but you can still tell that there is a cytoplasm inside of a cell membrane, and that there's always a dark dot corresponding to where the nucleus is. Each of these cells, these squamous epithelial cells, also have all of the other organelles like Golgi bodies and endoplasmic reticula, ribosomes floating around, but they're all too small. You cannot see them. The only features that you do know are there are based on the staining with methylene blue. So hopefully here you're able to capture one of these drawings and illustrate and label our nucleus, DNA, cytoplasm, and cell membrane. You notice that these cells might be a little bit different in their shape or different in their size. That's going to be something that we tackle a bit later. And occasionally they have these dark circles or squares or clusters of what looks to be maybe grapes together. Those dark circles are actually tiny bacteria. And so this gives us an idea of the size and scale difference between eukaryotic cells, like our human cheek cells, and the tiny little bacteria. There can be hundreds of little bacteria on any one of your cheek cells. Because yes, bacteria do indeed live in your mouth. You can also sometimes spot food if there was a meal recently had, so those will not stain as darkly or will have very different shapes. But ultimately, this is what we're trying to visualize with our staining. If you have the time, you are welcome to go ahead and try to visualize cheek cells without any stain. Odds are that you'll just see shadows and really not that very many cells. So let's take a look at some of our other animal cheek cells, because I think it's interesting to see how variation across the phylogeny or evolutionary relationships look. Some of the animals that we picked are mammals, and some of the animals that we picked fall within the reptile lineage, a particular reptile lineage known as the avians. And now, since these are all animals, we do expect them to be fairly similar, but I always like to give a little bit of phylogenetic context to understand how we expect similarities or differences to perhaps correlate. Now, we already looked at humans. Humans are sister to two other groups that we're going to be looking at, as both of these are mammals. We have a cat sample and a dog sample. Cats and dogs, respectively within Feliformia and Caeniformia, are more closely related to each other than they are to humans. So my naive hypothesis would be that whatever size ratio we have between mammals would be more similar between cats and dogs than it is between dogs and humans. There is also the issue of overall body size. And in this way, scientists can start to come up with hypotheses, come up with evolutionary correlates, and see long-term trends in anatomical evolution. As our outgroup to this, because it does benefit us to look at something quite far away, we have a turkey. So my naive assumption would be that whatever ratio there is between nucleus size and cell size, that there will be more similarity between and across mammals than there is to the turkey. Let's see how this plays out. But first, let's go ahead and take a look at what these cheek cells look like. Perhaps they look totally different. But spoilers, they don't, because ultimately we are all mammals and these are all made of squamous epithelial cells. Here we have a very pretty image of a couple of cat squamous epithelial cells. You see that there are a couple of bacteria placed atop here on this cell, and the nucleus is quite visible. We see something very similar up above, and then we see a cell that has some folds. Perhaps it was more than one cell, but this is a quite gargantuan one that seems to be folded a little bit. How does this compare to our turkey up next? Well, we again see a cell membrane, a nucleus, and there are some bacteria, or at least some sort of white dots. They did not stain, they may be different, So they're not the exact same bacteria that we see in cats, dogs, and humans, but they do have some pockmarks on them, suggesting that their mouths are full of bacteria, as we would expect for organisms that do not regularly brush their teeth or have a great cleaning system. Bacteria, going to be everywhere. And last but not least, we have a very particular dog breed, a beagle. We notice here that their cheek cells, at least naively without other focus, seems to be a little bit larger. But we do see the nucleus, we do see the cytoplasm, we do see the cell membrane. And now I'll give you a moment to go back, take a look at each of these, and draw them within your lab. So now hopefully you've had a chance to draw your samples and read through the next section on fluorescent stains. Because we're going to take a look at some images under the fluorescent microscope. Now in the lab, the reading has you discuss two different stains, propydium iodide and 4,6-diamino diaminodino 2-phenylindole, or DAPI. That is quite the mouthful of chemicals, so instead we'll use PI for propydium iodide and DAPI for the other one. Both of these are useful in labeling DNA. They preferentially bind to AT bonds, so as long as there is adenine and thymine bonds in your DNA, DAPI and propydium iodide, or DAPI at least, will preferentially bond there. Now, through your reading about DAPI and propydium iodide, you hopefully have figured out some of the differences between the two and what DMSO, or dimethyl sulfate, can help with. Particularly go explore the Wikipedia page on dimethyl sulfate to see what happens if it does contact your skin. But let's take a look at some images generated by using these stains. One from our lab, and the other is a very common image that you'll find on Wikipedia, taken with a confocal microscope. So a little bit more powerful, a little bit more focused, a little more pretty. But here we have our propydium iodide picture. Now, as the reading mentioned, propydium iodide is very good at labeling cell walls, and that's what we are looking at here. This is a onion skin cell, but I've done something different to it. Rather than just leaving it completely intact, I took a toothpick and tried to stab several cells. Now I took the smallest toothpick I can so that way we could maybe get some variation in spots. And at 400x you can see that there is a little bit of variation as to what cells were impacted. Propydium iodide cannot infiltrate the plasma membrane. It needs lots of other things to help it get through. Or we can kill the cells and puncture those membranes in the first place. If the membranes are dead, you should be able to see the nuclei. And in some of these cells, that's exactly what you see. The cells that do contain nuclei in this onion skin are clearly the ones that are dead. And as mentioned in the reading, this helps us identify potentially necrotic or deceased or apoptosis cells in different tissues. Now, this is something that we would not be able to figure out without under pretty much any other condition. These are very tough questions that stains help us analyze. Stains can also help show us the complexity of cells. And so in this image, you have three different stains. The red and the green are two different cytoskeletal stains, and the blue is DAPI. DAPI clearly binds to only the nucleus, as you see it preferentially found in the nucleus of these animal cells. But you can see how multiple different stains can help visualize the overlay in cell features. Plus, they look real cool, right? And now for the last portion of the lab today, and probably the most significant, because again, I want to make sure that you have it down as to how you can use a scale bar and measure cellular features or cell phenology with all the tools at hand. So the last part of the lab is asking you to calculate ratios of cell size to nucleus size, giving us an idea of how big the cell is versus how big the nucleus is in different species. This was performed in the biotechnology lab, lab 5, so please go ahead and check the details on that if you need to. But the general idea is that we're going to be taking images from the same magnification of a scale bar, our micrometer, and the cells themselves, and using those pictures alongside a ruler, and I do have a ruler in hand here, to calculate how big and what these different ratios are. So let's begin with our human cell. Change the color to red here. If I zoom in at all, this messes everything up. You want to keep the zoom scaled to exactly how it was. But what I am going to be measuring is this distance here, because I know that this image was taken at 400x, as well as this image taken at 400x, and while I couldn't ever pick up this ruler and move it to the top of the cell to see how big the cell is in terms of micrometers, I can place a ruler on top of it, and that's what I'm doing right now. So this distance I see as exactly 13.5 micrometers is the same as a tenth of a micrometer in real life. Going over to our cell, I'm going to take the longest axis as the length and the angle perpendicular to that as the width. So for the length, I measure, cell length, I measured exactly 10 millimeters. Oops, this should be millimeters is 1 micrometer. And then for cell width, I measure 7 millimeters. Now with these numbers, you of course can do your calculations. Set your apples to apples comparison. For my cell length, it would be a 10 millimeter comparison. Gives me how many micrometers, and you can solve for x. And all you'd need to do is change the numerator here, 7 millimeters, to give you the X for cell width. Obviously, you're going to be doing a lot more of these calculations, so please keep in mind that the reason I'm asking you to do this is at this point you should be more proficient with them, because the calculations are just a means to an end. What we want to do is actually do our comparisons, see if there are differences. The other thing that we need to measure that is very difficult to measure is the nucleus itself, that little feature. So what I will do to help this along, at least on the iPad itself, is I will circle the nucleus, that being its length, that distance being its length, and that distance being its width. Zoom out again to retain the same size, the same fit. And I'm seeing something like nucleus. We'll change pen here because get a little bit too small to read. So nucleus length as 2.5 millimeters and nucleus width as 2 millimeters. And again, you can go back and calculate each of these different metrics. Once you have those metrics, you can take length times width. Let's make this a little bit thicker. You can take length times width to calculate an area. And what I want you to do in this lab is compare cell area to nucleus area in dogs, humans, turkeys, and cats. Once you can get this value down to a single number, you can then take the cell area for, or this ratio for one of our species. Let's say that we first calculate it for humans. You can then take that whole ratio and divide it by the ratio of dogs, cell area to nucleus area, and therefore you will get a comparison of these two. With a single numeric metric like this, you can then make comparisons between all 4 of our different species. So I'll get back to our regular zoom here and show you what the scale bar and the cheek cells look like for our dog, what the scale bar and cheek cells look like for our turkey, and what, just those 3 actually. I didn't include the cat because I figured it's actually a significant amount of work to do these calculations, but I forgot that I didn't include the cat. Now, just because these pictures are not on your phone does not mean this does not work. You can take these measures directly from the monitor or phone that you are looking at. The sizes will be different. The simple measures of millimeters will be different depending on the screen, but the ratios, the aspect ratio, will always be the same. So despite the fact that your numbers in millimeters might be different than my numbers in millimeters, the ultimate ratios will be the same. And so from this lab, and when we get to the practical, I will be asking you to compare ratios of things. This means that you need to show me all of your work, and this means that you need to be able to whittle down these large numeric comparisons into single metrics to see how these ratios differ. On the practical, I'd expect that you'd be able to do this for multiple cells in a field to see what the size differences are, perhaps between cell length and nucleus length across different cells in the same field of view or different species. So please make sure that you're able to calculate these independently for ratios of length, of width, and of area. You have your images of humans, dogs, and turkeys to help to get you to finish the assignment. Good luck!